Too long, so many years.
About 25, on my reckoning.
Crazy.
Back in 1977, a Nature paper was published. This is testament to Saxel that determined serial passaging and differentiation of myogenic cells, a model of muscular dystrophy. It was published in 1977, and 13 years later, around that time, Dr. Eric Olson was unveiling the genetic program. And by the way, Deepak, I took these pictures this morning.
Is that right?
I'm doing what we used to do in collaboration with a faculty member that reports to me. He's studying autophagy throughout this differentiation program. So that's kind of cool. So by way of introduction, Dr. Srivastava is an MD. He's president of the Gladstone Institutes and director of the Roddenberry Stem Cell Center. He's also a distinguished professor in pediatric developmental cardiology at the University of California. In addition to caring for sick children as a physician, Dr. Srivastava runs an active research group that studies the biology of heart development and regeneration. It's so great to see you. So Dr. Srivastava, thank you for joining us. Take it away.
Great, Jeff. Well, I'm delighted in this new Zoom world we're able to do something like this. Jeff and I were postdoctoral fellows together in Eric Olson's lab more than 25 years ago, and we haven't had a chance to intersect so much. So I'm delighted to be here with you all today. As Jeff said, I am a pediatric cardiologist, and when I had moved from Boston, where I did my clinical training, to train with Eric, and when he was at MD Anderson in Houston at the time, we set out to try to see if we could understand how cells are fated to become a heart cell, much like what had been discovered with the myogenic factors in skeletal muscle. And we were fortunate we were able to find some key factors that were important for heart formation, which at the time we knew almost nothing about. But at that time, we weren't able actually to quite figure out a MyoD equivalent in the heart, if you will. And I'll share with some of you about that in our more recent work. So Jeff, I'm going to try to share screen here on my messy desktop. Let's see here. Okay, I think I've got this. Oops. Okay, there we go. Great. So what I thought I'd do today is tell you a couple of stories, but they all sort of center around the questions: can you, if you understand the state of a cell well enough in normal states or in disease states, if you understood what goes wrong in that cell well enough, might you be able to reprogram that cell so that it does what you want it to do? And there are many incarnations of that that are potential. So I'm going to touch on that with a couple of different stories. But all of them sort of center around a common theme, and that is, in order to understand the system well enough to reprogram it, you have to first understand comprehensively the gene networks that are at play, both in homeostasis and in the setting of disease, in order to know what and where to correct. And so a lot of our efforts are focused on that fundamental question. And we've channeled it in a couple of different ways, both in the cardiac regenerative medicine space, as well as in intersecting with human genetics. But the basis for both of these branches really is in a deep understanding of the developmental biology of the organ, because that's when so many of the gene networks are laid down that dictate fate and that go awry in the setting of human disease, even if that disease occurs later in life, as I'll mention. And so our laboratory has invested a lot of effort over the years in understanding these developmental networks. And I won't have time to talk about it today, but actually I'm very, very excited about the new opportunities and understanding developmental biology at a deeper level that's come through single cell technologies, whether it's single cell transcriptomes or chromatin landscape through ATAC-seq. Now, more recently, single cell approaches with the proteome. But together, we'll have the opportunity for the first time to break down the individual cells that are involved in forming a three-dimensional organ and asking which even a small subset that goes awry, might that cause a developmental defect? And actually, that's what I'm most passionate about, because the children I take care of are often born with heart anomalies where probably a tiny fraction of their cells in their heart went awry and the rest of the heart was pretty normal. And that's what we've got to figure out, and we've never had the tools until now to really understand that. But the way we've been able to channel this information, as I mentioned, is in the cardiac regeneration field, and is either through harnessing the resident cell types that are existing mostly in the heart, which are cardiac fibroblasts and cardiac myocytes in different ways. And let me share with you that aspect first. And in this space, the problem that we're trying to address is in part illustrated here, which is on your right, a section of a heart with the left ventricle on your right and left ventricle here on your left. And here a heart has had a coronary occlusion event, like what might occur in a heart attack. And the downstream muscle supplied by that vessel has died and is replaced by scar tissue that you see in purple here. And this scar tissue is helpful and it keeps the heart from rupturing, but it's not muscle, it doesn't contract, and the heart has little to no capacity to regenerate itself after injury. And so people are left with this, and this leads to heart failure, and ultimately at the end stage of that is a transplant, because we really have no therapeutic approaches right now to try to address the problem here, which is you've lost muscle, and so you might want to gain muscle somehow. So we've taken two strategies in the last many years to try to create new muscle. And these other groups are trying to inject stem cell derived cardiomyocytes into this region. And I think there's some promise there. There are many hurdles to overcome, including getting those cells to integrate with their neighbors and contract in synchrony, et cetera. The approach we took was ask, could we do something with these cells that are already in the heart, the fibroblasts and the cardiomyocytes, and use them to generate new myocytes? And the easiest of all approaches would be to get the post-mitotic cardiomyocyte that's there in the adult to somehow go back and be able to re-enter the cell cycle and divide again and make more of itself. That's the simplest approach. Of course, there have been people who've been trying to do this for decades, and it's been challenging in part because even when you could get a cell to divide, an adult cell that's post-mitotic, when it did divide, it underwent mitotic catastrophe and killed itself. So unless it goes through the cell cycle in a perfect manner, this doesn't work very well. And so a couple of years ago now, we reported success in doing this with a combination of 4 cell cycle regulators that work in tandem. So CDK4 and cyclin D, which work together at this phase of the cell cycle, and CDK1 and cyclin B, these 4 together, but no 3 alone, could get a cell to go all the way through the cycle and then stay alive. And we could visualize this under live microscopy and see the cells divide. We also utilized a variety of genetic tricks where we could genetically label a cardiomyocyte, and using a variety of tricks I won't go into, definitively demonstrate in vivo whether that cell didn't just undergo karyokinesis, which is not so hard, but actual cytokinesis where the cell divides. And by doing that sort of a genetic approach, we were able to demonstrate that, in fact, about 15 to 20% of the cells that had all 4 of these factors could successfully divide and stay alive. In mice, when we did this after a myocardial infarction, it increased cardiac output. And so this looked like a really great way to deal with the problem. We've run into so far a problem, and so I won't say much more about this, which is a delivery hurdle. While this is great from a scientific standpoint, from a translational standpoint, the notion of delivering cell cycle regulators with a gene therapy, a viral approach, which is what we had done in mice. And now one of my former fellows, Tamer Mohammed, who runs his own lab, has done in pigs, is really not probably a viable approach because of the oncogenic risk. And so we're looking at a variety of ways, including a modified RNA and direct protein delivery. And in fact, we had tried modified RNA some years ago, but it didn't work very well and was actually quite toxic. But we're encouraged now by the Pfizer and the Moderna vaccines that are injected into the muscle and clearly are able to make spike protein pretty efficiently. And so they have new technology for packaging the modified RNA and lipid particles. And so we're now trying that again. And if that works, then I think this is a very, it would be a great delivery, because modified RNA the problem is it only lasts maybe 3 or 4 days, but that's exactly what we need here, because you don't want these around for much longer. And within the cell cycle, cell division occurs within 3 or 4 days. The second approach that we've taken, that we've invested a lot of time in and effort over the last decade, has to do with this pool of fibroblasts in the heart. And it turns out that the heart, while you think of it as all muscle, in fact, half of the cells in your heart are not muscle, but rather they're fibroblast cells. And these play an important architectural role in the heart. They secrete important signals to the muscle, and they're also the ones that go and make the scar that you see here in this heart. And years ago, we had asked the question, could we reprogram these cardiac fibroblasts, they're right where they are in the heart, into something that looked like a cardiomyocyte? And this is akin to the MyoD experiment done beautifully by Hawantar's group in the late 80s, where MyoD, they showed, could convert a skin fibroblast into a skeletal muscle cell. And it turned out over the years that we and others had tried to do this with single transcription factors as we discovered them, but none of them were sufficient for this conversion. And inspired by Shinya Yamanaka's work of combinatorial factors, reprogramming fibroblasts to stem cells, and Shinya I had recruited to Gladstone back in 2007, so his lab was neighboring ours on our floor. We asked, maybe in the heart, we've been looking for a single factor, but in fact, we need a combination. And so we did a screen with about 20 different transcription factors and finally came to the observation that a combination of these three, GATA4, TBX5, and MEF2C, together were sufficient to push a cardiac fibroblast to what we called an induced cardiomyocyte state. When we did this on plastic and a dish, it worked, but it was pretty inefficient and incomplete. However, when we did it in vivo, which was our goal, in a mouse heart, in its natural milieu of the ventricular chamber, the reprogrammed cells that we could label genetically and trace were very, very similar to adult ventricular cardiomyocytes. They electrically coupled with their neighbors, so they beat in synchrony. And by MRI that we can do in these mice, we could actually significantly improve the cardiac output of these hearts. So that was great. And we have spent a considerable amount of time trying to think about and analyze the basic biology of this. Really, what we found fascinating event, that you could have this wholesale epigenetic shift from a fibroblast to a myocyte stage that leads to this phenotypic transformation. And I won't go through the details of this work that was published a little over a year ago, but I'll just share a couple of interesting observations. To try to get at the mechanism, what we did is we utilized single-cell RNA-seq approaches so that in vitro, when we do this conversion, we could separate out converting cells and non-converting cells. We used a TAC-seq so we can ask the question in a temporal fashion over time, how does the chromatin open and close to help execute this shift? We use ChIP-seq to ask, where do these transcription factors actually sit on DNA and in what combinations? And then we utilized a machine learning approach in collaboration with Katie Pollard's lab, a computational biologist, to synthesize this information to learn something. And I think the most important things we learned for here is schematically shown here, and that is that in some cases we found that a single reprogramming factor, when introduced by itself, couldn't really do anything. But when all three were together, they worked in a cooperative fashion, actually could open up closed chromatin and activate gene transcription of cardiomyocyte-related genes. There are other cases where a single factor actually could do something by itself, and it functioned more like a pioneer factor of opening closed chromatin, and then by virtue of then recruiting the other factors, could then enhance activation and stabilize that at certain sites. And then perhaps most interestingly, we found that at some sites, an individual factor could go sit on DNA and function like a pioneer factor and open up chromatin. But it was opening up and activating the wrong things. So none of these three factors are actually completely specific to the heart. GATA4, for example, is present in endoderm, TBX5 in limbs, MEF2C in the brain. And so how is it that these three together result in specificity of cardiomyocyte gene expression? And it turns out that when we put in a single factor like MEF2C, it would activate actually neuronal genes. But when the other two were present, then they would help refine this and actually remove that single factor from these aberrant loci that you didn't want to turn on in the heart and actually shut that off. And so the combination not only cooperatively guided cardiomyocyte transcription, but cooperatively repressed transcription of unwanted genes in that particular lineage. So we think this is going to be a general paradigm for how reprogramming factors work, not just in this setting, but in many other settings as well. Now, on a more translational front, we obviously wanted to move this not just in mice, but into human cells. And there it turns out that the same three factors did not work in human cells. But if we substituted GATA4 for another transcriptional co-activator called myocardin that actually works with MEF2C, then together these three factors were sufficient to turn a human cardiac fibroblast into a pretty nice looking cardiomyocyte, like one shown here with these beautiful stripes that represent the contractile sarcomeres in the cell. So that was good. We had a combination that worked in human, and I should say also in pig cells. And we actually devised a novel adeno-associated virus, or AAV, that had greater tropism to human cardiac fibroblasts and pig fibroblasts in order to deliver this more efficiently. And so we took this into a larger animal to ask, can we reprogram well enough in a thicker heart, more like the humans, in a meaningful enough way to actually make a functional difference? And so we've now done this in pigs, like what you see here is a pig heart. Down here at the apex, you might appreciate that this area is more blanched, less red than around here. This is the area of the myocardial infarction induced by a coronary occlusion event with a balloon inflation in the coronary artery for about an hour and a half. So we directly into the muscle with these adeno-associated viruses injected these three transcription factors. And I'm showing you a section here of analysis of this. And what we did not have in the pig that we had in mice was we didn't have the ability to put a genetic, GFP or some other marker on the fibroblast and lineage trace them to ask rigorously, does a fibroblast actually turn into a muscle? But we did the next best thing here is, what we did is we co-infected these hearts with a retrovirus encoding DS-RED. And retroviruses, you may know, only infect dividing cells. So myocytes are never dividing in the adult. And so we surmise that if a cell was red, that means it was dividing at some point, and so it should be a non-myocyte. And so if a red cell now has sarcomeres, we would make the assumption that perhaps this was a reprogrammed cell that came from a non-myocyte. And so here in this one section, I think you can easily appreciate on the left a number of red cells that are long and large. And on the right, if you just look by this in the white channel, gray channel, I think you can start to appreciate that these labeled cells do in fact have sarcomeres by confocal microscopy. And so this would suggest that they are in fact reprogrammed cells, and they're quite abundant number of them. And so at this point, we had transferred the technology into a startup company called Tenaya Therapeutics that started a few years ago and has raised a series of rounds now to try to help take this technology and reduce it to a therapeutic and get it to a clinical trial. And so they're doing really well and are on their way. And I'll just show you one slide from them, with their permission, that of a preclinical trial they've done in pigs. They're about 10, under 10 in each group. And in this case, they've created a injury, waited about a month so that the decrease in the ejection fraction of the heart was stable, and then dosed these pigs with the gene therapy approach for these reprogramming factors. And what we're measuring here then is in each pig, the change in ejection fraction over time by echocardiography. And the control pigs, you can see they don't change very much. They've already had their injury and they're pretty flat. In contrast, in the pigs that were treated, after about 2 months, we have on average about 11% increase in ejection fraction. And the absolute numbers here go from somewhere around 30% to 40%, which is enough to take somebody who might be on a transplant list, who wouldn't need that. They're not going to still be able to run a marathon or even a 5K probably, but it's of great clinical significance. So we're excited that this is on its way. Still a number of hurdles to overcome to really be able to do safety testing, toxicity, and really get this to work in the simplest possible product. But I think it provides an example of how, if you understand the system well enough, you can reprogram cells to do what you want them to do for a therapeutic benefit. Now, I want to share with you a story that's not published, that hopefully will be published very soon, led by Mickey Alexanian, who's a very talented postdoc in the lab currently, where he's asked a different question, and that is these fibroblasts that are in the heart already and get activated after injury and go and lay down this scar. Could we do something to change their scarring role in times when actually that function is maladaptive and is not helpful? And in the particular case where that we know is an issue is in the setting of heart failure, where there's a damaged heart for whatever reason, and over time that heart gets worse.
Deepak?
Yeah.
I see Mike Johnson has his hand up. Dr.
Oh,
Johnson.
good. Yeah, I can't see everybody, so please go ahead and interrupt. Feel free to interrupt.
Doctor, I just wanted to say thank you very much for this excellent presentation. I have one question, and I know you're transitioning into heart failure, and some of your slides have focused on cardiac remodeling with an improvement in overall cardiac output. My question to you is, what portion of the cardiac output is improved? Is it the stroke volume, or is it the heart rate, or what is it?
That's a great question. It is the stroke volume, so the actual amount that the heart is able to squeeze with each beat.
Sure.
Checked out. That's right.
Okay.
Yeah.
And you had mentioned ejection fractions between 30 to 40. Could definitely be a sizable difference in getting that individual or that specimen back in the game again. Absolutely.
Yeah.
Well, thank you very much. I appreciate it.
Yeah. Thanks for your question. And so the scenario I illustrated before can lead to heart failure. So can many other conditions. And in that failing heart, what we do know that it gets worse over time, and there's a lot of fibrosis that occurs, and we haven't fully understood how the pathology occurs that makes things worse. But what we have known is that there is a shift in the transcriptome of the whole heart in ways that are likely maladaptive. And And some years ago, a colleague of mine, Sap Halder, who's also at Gladstone, had done some beautiful work that suggested that there's signaling through the chromatin that is key in this transcriptional switch, if you will, that was maladaptive. And what he had shown is that bromodomain proteins sit at the center of this maladaptation. And bromodomain proteins, which are co-activators, and BRD4 is one of the family members, can sit in enhancers and activate transcription. And what we knew is that under conditions of stress, that these bromodomain proteins could collect at stress-activated enhancers and turn on a whole set of genes that were actually hurting the function of the heart rather than helping. And he had also shown that using a small molecule inhibitor of BRD4 called JQ1, which is a tool compound used to competitively displace BRD4 from enhancers, that in fact you could improve the function of the heart. But what we didn't know is what was the mechanism of this, where was it sitting, and what cell type even was this working on? Is it myocytes or something else? And so we had done an experiment shown here where if you induce stress in the heart with something we call thoracic aortic constriction, so you just tie a suture around the aorta and increase the pressure that the ventricle has to pump against, you can cause the ejection fraction, the function of the output of the heart, to go down. And we found that if you treat with JQ1 these mice, then the function goes back up. But then if you stop treatment, it actually goes back down, so it's reversible. And then not shown here, if we treat again, even in this late stage, the heart function goes back up. So it can toggle back and forth. And this suggested that something was going on perhaps outside of the muscle, because we wouldn't expect that to toggle so efficiently. And so Mickey decided to do a very large experiment, taking advantage of single cell biology that might get to allow us to sort out which cell types were at play. And so here what he did is he put these hearts under stress, waited a few weeks after they had started to have heart failure, treated them or not with this JQ1 compound, waited a month, and then stopped treatment in one treatment group and continued in another. And then we disassociated these hearts, took the cardiomyocyte population and then the non-cardiomyocyte population. And I won't show you the data, but it turns out that what we found is in the cardiomyocyte, there was very little transcriptional change at all. So we didn't see much change there. But in the non-cardiomyocytes, we did single-cell RNA sequencing and single-cell ATAC-seq to look at the chromatin changes. And we did it in each of these 4 conditions that I mentioned, and did both of these approaches with each of the same hearts. Okay, so I'm going to show you some data from that I think was very revealing. And we looked at all the cells, but for sake of time, I'm going to focus on the main population where we saw the biggest difference, which turned out to be the fibroblast cells. And what we saw is if in green here are the cells from the sham, and these in these UMAP plots, each dot, of course, represents the transcriptome of a single cell, and the location of it represents its one cell similarity to another. So upon TAC, or this pressure load, cells moved from this green state to this red state and were very different transcriptionally. When we treated with JQ1 in blue, they got reverted back closer to the sham controls, so they look like they got restored. And when we withdrew TAC in orange, they shifted back towards the stress condition. So that was interesting. And I'm showing you here on the right a marker gene of what we know of as what we call the activated myofibroblast. This is the fibroblast that goes from the quiescent state to this active proliferative state that's laying down lots of collagen and is profibrotic. And so what you can see here is JQ1 prevented activation of periostin and brought cells back here in this age. And when we stopped JQ1, periostin got reactivated. And that's quantified here on the right. So that suggested that by inhibiting BET proteins, we could maybe toggle the fibroblasts back and forth from this activated state to an inactivated state. And then to ask, I'm not showing you, but there is a host of other genes that were all consistent with this going from an activated myofibroblast to not back and forth. So we asked, how is this actually controlled transcriptionally? And to that we turned to the ATAC-seq data. And because in this system, I mentioned to you, we had done ultrasounds at each stage of this process, we could correlate ejection fraction with these different states and ask which chromatin states were going, say, from a closed state to an open state when the ejection fraction went down, closed again when the ejection fraction went back up with treatment, and maybe opened again when we withdrew the drug, and the opposite as indicated down here schematically. And so we analyzed the ATAC-seq data in a graph that looks something like this, where we looked for a correlation of things, enhancers that open when ejection fraction went down, or enhancers that closed when ejection fraction went down. And you can graph this looking at the p-value and the degree of correlation of opening and closing with function. And I won't go through all the data with you, but point out that one of the most anti-correlated loci was nearby a gene called MEOX1 that encodes for a transcription factor that's not been studied very much as yet. So here we've got a transcription factor that's highly anti-correlated with this situation. And when we looked at the expression of this transcription factor, it, very similar to periostin, was getting activated in the stress setting, would get shut off with treatment, and would come back on if we withdrew treatment. So that, again, correlated with the activated myofibroblast state. And so we did ChIP-seq with this transcription factor to ask, where does it bind to DNA and what might it be doing in these fibroblasts? And it turns out that the top Go terms are shown here, which are all indicative of this activated profibrotic state of the fibroblasts, turning on things like CTGF, periostin, there's integrin, that are all well-known things that are involved in fibrosis. So that suggested that perhaps this transcription factor may be a key switch, if you will, of a fibroblast going from a quiescent to an activated state. And so we've done some experiments to ask if that is in fact the case. And what I'm showing you here is a ProSeq experiment where we're looking at the nascent RNA that's being transcribed, both from coding regions, but also this approach allows you to see enhancer RNAs that tells you if an enhancer is actually active. And because we had done ATAC-seq, we knew all the peaks or the potential enhancers nearby the MEOX1 gene that might be getting activated to turn on this transcription factor. And for this experiment, we turned to an in vitro system where we knew that if you stimulate fibroblasts with TGF-beta, they actually undergo this shift to an activated myofibroblast. And here, what I think you can see is that when we add TGF-beta to these fibroblasts, there's an enhancer RNA region that gets activated nearby this MEOX1 gene. And so we suspected that perhaps this could be the key stress-activated enhancer that's responsible for turning MEOX1 on. And because of beautiful CRISPR technology that's now available, and Jennifer Doudna also runs a second lab in addition to her lab at Berkeley at Gladstone, we were able to use a CRISPR-I technique for CRISPR interference, where we utilized a guide RNA directly to this enhancer RNA area and asked if we used CRISPR-I to this, would we now not get transcription from this enhancer region? And that, in fact, turns out to be the case, as you see here. TGF-beta stimulates transcription at this spot, And then when we use a CRISPR-I to this, we shut that off. And then we can ask the question, if we shut this off, this one stress-activated enhancer, is that enough to not allow MEOX1 to be turned on? And that, in fact, turned out to be the case when we looked at MEOX1 RNA levels in the setting of TGF-beta, it goes up. And with this CRISPR-I, we don't get that activation at all. So that points to this one locus as the key stress-activated enhancer to turn on MEOX1. And then finally, we can ask in the system now also, if you prevent MEOX1 from being turned on, now do you prevent this activated myofibroblast state from even occurring? And we have done that. I'm just going to show you one example for one of the key genes, CTGF. At this locus, if we stimulate with TGF-beta and a control siRNA, then we get activation of this CTGF transcription. However, if we use a siRNA to MEOX1, even with TGF-beta, we don't get this, and we see that across the board for almost all the pro-fibrotic activation that occurs. So what we think is going on is that MEOX1 is in fact is the key transcriptional switch for this activated myofibroblast state. Now, what I've shown you in the heart is not necessarily unique. MEOX1 isn't unique to the heart. And in fact, in many other organs in our body, fibrosis occurs in disease settings that we have no way to treat. And that's true for lung, like it occurs in idiopathic pulmonary fibrosis, liver, and in the kidney. And so we asked, in human fibroblasts from each of these tissues, might the same thing be going on? And what I'm showing you here is that when you stimulate any one of these with TGF-beta, it turns out the MEOX1 expression goes way high in each of these settings. And in all of these settings, we can actually also reverse that with JQ1. So we think that this may be a universal switch that we've uncovered for this activated myofibroblast state. So just maybe schematically to summarize what we think is going on here is that in a normal heart or in other organs, there's a fibroblast pool that's quiescent and it's doing what it should be doing. During times of stress, in a BET-dependent manner, specific enhancers get activated that are stress-induced that result in activation of MEOX1 that then serves as the switch to turn on a host of pro-fibrotic genes typical of an activated myofibroblast. And so if we could interject in a specific manner at one of these steps, we could actually alleviate this problem. And in fact, now we've used CRISPR deletion to delete out this small enhancer in mice. And now we're in the process of in vivo stressing those mice to see if even under conditions of stress in multiple organs, we actually prevent this maladaptive myofibroblast activation and the subsequent fibrosis. So we're very excited about that as a potential therapeutic as well, by getting to a very precise mechanism by which this transition occurs. So those are the stories I wanted to share with you on the sort of approaches to take the resident cells in the heart and make them do something that they don't normally do to address human disease. Let me pause there and ask you any questions. Otherwise, I'm going to move on to some of our work on cellular reprogramming as it relates to human genetics of disease.
There is a question.
I'd like to ask a question if I could,
Sure.
please.
Go ahead, Dennis.
Did you ever look at spironolactone on this process?
Yeah, it's a great question. So I think that question is getting to the fact that spironolactone is used in chronic heart failure. It has been for many years, based on a trial done probably 2 decades ago that showed that for reasons we don't completely understand, that slows the progression of heart failure over time. So it's a very interesting question, whether that might actually affect fibrosis. We haven't looked at that, but it would be very interesting to look at because the actual mechanism of that helpful effect is still not completely clear. It's a good thought.
go ahead.
All right. So, our laboratory has had a longstanding interest in trying to ask, are these key developmental networks that we and others have discovered over the years, are those in fact the ones that are mutated and hit in the setting of human disease? And can we use that knowledge to then understand mechanism and get to potential therapeutics? And so I want to share with you, we've published a body of literature illustrated on the last slide in this area, but I wanted to share one story with you that I think has now gone all the way from identifying a genetic cause to having a potential therapeutic in work that we just published last month. And this story is now actually embarrassingly about 18 years old. It started with a family that I took care of that had this disease here, it's called calcific aortic valve disease. It's the third leading cause of adult forms of heart disease. It's an age-dependent disease for reasons we haven't understood clearly before. But over time, people develop more and more calcium deposits in their aortic valve that separates the left ventricle from the aorta. And ultimately, it looks something like this, where it gets very brittle and doesn't open and close well, and ultimately needs to be taken out surgically and replaced with a PIG or mechanical valve. And this can happen in the general population and in an age-dependent fashion, but we do know that there's a condition where people are born with an aortic valve that doesn't look like this with 3 nice leaflets in this Mercedes sign, but rather they're born with only 2 aortic valve leaflets, and we call this a bicuspid aortic valve. And it turns out this is actually the most common of all birth defects because it occurs in 1 to 2% of every person born across the world. So that's a lot of people. So it's very common. However, most people don't even know they have this when they're born or until they're 40 or 50 years old when this occurs, and then they have trouble and have aortic valve stenosis. And so but the problem probably was initiated early in life with this birth defect that then manifests over time. Sometimes children actually do, even when they're first born, have problems when this valve isn't just bicuspid, but it's also very thick and doesn't open and close well, and they actually need a procedure as soon as they're born. And that's actually how I, as a pediatric cardiologist, came across this family that you see here when I was in Dallas. And I took care of this little boy here at the bottom who needed an intervention in his first few weeks of life. But when we took a deeper family history, it turned out that across 5 generations in this family, they had had trouble with this valve, and mostly with people in their 20s, 30s, 40s, or even 50s, where they developed calcification and needed a replacement. So many people had surgery, and many had died prematurely in this family. And so we were able to, because there are so many people affected across many generations, we were able to do linkage analysis back in the day and identify a single gene mutation that resulted in this autosomal dominant transmission. And that single gene mutation affected a famous gene that I'm sure all of you are familiar with called Notch1. And it was a heterozygous loss of function mutation on one allele that was enough to cause this disease with 100% penetrance in this family. And now we and others have identified many other families with this condition. So that was great. We, for the first time, knew a genetic cause of this very common disease that has no medical treatment. But that didn't really, knowing the genetic cause doesn't really help you unless you understand mechanism. And so we immediately, of course, looked at mice that were heterozygous for this, that had, of course, been made, because Notch1, as you know, is important for virtually every organ in the body, some way or another. And it turned out that Notch heterozygous mice were just fine, but the Notch homozygous mice died in early embryonic development from failure of vascular development. And so it died by embryonic day 9.5. So that wasn't very helpful. So we were stuck for a while, and then fast forward a few years, iPS technology came around to induce pluripotent stem cells. And as I mentioned, Shinya Yamanaka is our colleague on the floor. So we immediately made iPS cells from these family members. And this was work led by Christina Theodoris, a MD PhD student who's now a pediatric genetics resident at Boston Children's. And she undertook this project and utilized iPS cells to try to understand mechanism. And I'll share with you in the next few slides what she learned from this, which I think beautifully laid out for us the biology that was going on. And by using iPS cells and then turning them into endothelial cells, which we, from other work, knew was the culprit cell type, she grew billions of cells and asked, where does NOTCH sit on DNA? How does that change the epigenetic landscape and subsequently the transcriptome? And I'm just going to, since this work is published, summarize schematically for you in a few slides what we learned. So what we learned is that NOTCH, by virtue of sitting on the cell surface, actually is in a position to sense the blood flow across this valve and in ways that take advantage of this shear stress that it experiences. Its normal function is to then, translocate to the nucleus upon shear stress and activate a whole anti-calcific gene program that essentially represses an osteogenic fate and represses a number of inflammatory signals. That's its normal job. In the setting of haploinsufficiency or heterozygosity, there's a reduced amount of NOTCH that's doing this, and as a result, you get aberrant activation of a whole host of pro-osteogenic and pro-inflammatory signals. There are about 1,000 genes that we found in the system upregulated. And because we had looked at the epigenetics and the ChIP-seq with NOTCH, Christina was able to reconstruct a whole gene network at quite significant depth and understand how this actually occurs. And it turns out from that analysis that we found that even though 1,000 genes were dysregulated, fully 800 of them, or 80%, were activated simply by the upregulation of just 3 key transcription factors shown here: SOX7, TCF4 that mediates Wnt signaling, and SMAD1 that mediates BMP signaling. And together, these 3 were accounted for most of the gene dysregulation in what appeared to be essentially a cellular reprogramming event where a cell went from an endothelial cell to now behave more like an osteoblast cell and do what a good osteoblast should do, which is to basically lay down calcium. So that's what we think is actually the crux of this disease, is that normally NOTCH should prevent this osteoblast cell fate in the valve. And there are a host of reasons we can talk about why the system is set up this way, because during development, a valve has many characteristics of chondrocytes or osteoblasts, but then the body appears to exert a system to then say, no, now stop, we want you to stay cartilaginous and not become bone-like, and NOTCH is the driver of that. And in the setting of haploinsufficiency, that's de-repressed, these cells become more osteoblast-like and lay down calcium. And it's as simple as that. That's the disease. Now, because I mentioned that this funneled down to just three key transcription factors, Christina asked, well, maybe this is a druggable system, and maybe we could find drugs that would actually correct the network and not allow this cell fate switch. So she designed a very clever assay that leveraged a technology that was new at the time called targeted RNA sequencing. So here she designed a system where we could assay not just 1, 2, or 3 genes with every compound. Here we screened a relatively small library of highly selected compounds, but rather we could ask how do almost 120 genes in this key network change with each drug by sequencing in a targeted fashion all 120 genes. And then Christina developed a machine learning approach to either classify normal cells or heterozygous cells as separate, and then ask, do any drugs now take a abnormal heterozygous cell and now classify this as a normal wild type cell because of the drug? And so we did actually find 6 hits from this screen that had significant correction of the gene expression overall, so that this algorithm was classifying them as normal cells. And I'll show you what that output looks like here in this PCA analysis. So what you're seeing here are dots, where each dot represents a condition. So The blue dots represent where wild type cells would be classified by this machine, by this algorithm. And the green cells that you see here are what a patient heterozygote cell would classify. And you can see that these separate very well. So there's almost no overlap. So we have very clean calling of wild type or heterozygous disease cells. And now red are dots that where the patient cells have been now treated. These are patient iPS-derived endothelial cells, have been treated with one or another drug. And what I think you can appreciate is most drugs don't do anything. They still cluster with the green cells. But now you'll see, every now and then you see a cell, a red dot that is now clustering with the blue dots. And so these are what we call our hits, because our algorithm is saying, we think this is a wild type cell by its transcriptome, but it's by virtue of that one drug. So that's great. We had some hits, but obviously these were in vitro and endothelial cells. And we obviously wanted to be able to try this in some in vivo fashion in the setting of a disease. And you might recall I mentioned that the mice that are heterozygous for Notch1 were normal. So, but this is an age-dependent disease. And so Christina had the idea that if this was age-dependent, then perhaps mice don't get the disease simply because their telomeres are longer than humans. And we've known that their telomeres are maybe 4 times as long as humans. And so she crossed the Notch heterozygous mice with mice lacking TERT, which is the RNA component of telomerase. And so when you do this in successive generations of breeding, the telomeres get shorter and shorter with each generation. And what she found, quite strikingly, is that if you just shorten the telomeres a little bit by the second generation, these mice completely recapitulate the human phenotype of aortic valve calcification and stenosis and nothing else. And what I'm showing you here in this cross-section like this through the aortic valve, here you see in a normal mouse, these are the 3 leaflets. They're thin and separable, and this is a close-up. And in contrast, in these mice with shorter telomeres, you see that this valve leaflet is very thick, like what occurs in humans, and with alizarin red indicating calcium, they have a lot of calcium deposits. So this is exactly what a human valve looks like in disease. And so now we also had an in vivo way to both test our drug, but also to ask the question that we thought from the in vitro setting of human iPS cells, that the disease problem here was a cell fate switch to an osteoblast-like cell. And so we have a way to test that now in vivo. And so we stained with an antibody to RUNX2, which is, you may know, is a key transcriptional master regulator of the osteoblast fate and marks osteoblast, and asked, do these cells in the valve now activate RUNX2, and are they more like osteoblasts? And in fact, that is the case in vivo, even suggesting that that is consistent with that being a disease problem. And so now we had this animal model to test these 6 hits. And over the last few years, we've tested all of these drugs in the animal models, and 2 of them actually seemed to work pretty well. And one of them that was actually the best network corrector, if you will, of shifting gene expression back, actually was the one that worked the best in vivo also. And I'll just show you that data briefly here. Here, what we're looking at is by ultrasound, the peak velocity of blood that goes across the aortic valve. And this is exactly what we do in humans to measure aortic valve stenosis, because by simple physics rules, if you narrow the opening of a pipe, then fluid has to go, same amount of fluid has to go faster through it, right? So that's what happens in the aortic valve. So you can see here, in these mice, it's incompletely penetrant, the disease, but you see a number of mice that have aortic valve stenosis as measured by this peak velocity. And the same was actually true for the pulmonary valve that I didn't mention, that happens both in humans and in this mice. And so now with one of these drugs, XCT790, we see that this is significantly reduced. And we have this one outlier, but overall, this is very effective. And the same was true for the pulmonary valve, as you can see here. So we were able to actually treat these mice with this drug. Data I won't have time to show you. We've actually also shown that it reduces the thickening of the valve and the amount of calcification or calcium deposits that occur in this. So we're very excited that we've been able to, by identifying the mechanism and the gene network disruption, screen for a drug in a very targeted fashion and test this drug in vivo in mice, that this, we have maybe for the first time a drug that might prevent and treat even this stenosis. Now, everything I've told you so far has had to do with the Notch1 heterozygous setting, either in people or in mice, but in fact, maybe I didn't mention, the family I showed you, like in most cases where there's autosomal dominant disease, is a rare type of setting. And if you took everybody who has aortic valve stenosis, it's only probably about 4% that where you can implicate Notch as the cause. And so the majority are going to have this disease without a Notch mutation, but the pathology we surmised would be very similar. And since we screened for this drug not on to target Notch, but rather the whole gene network that occurs in this sulfate switch, we thought that perhaps this drug might also work on people who had other causes of aortic valve stenosis, but the pathology funneled down to the same sulfate switch, which we think is going to be the case for almost everybody with this disease. And so we collaborated with the group in Russia that had happened to collect primary human aortic valve endothelial cells from explanted human valves at the time of surgery. And so they had they had 9 calcified trileaflet patients, 12 calcified bicuspid aortic valve patients, and they had a bunch of normal controls where for other reasons in a normal heart the valve was taken out or after autopsy. And so we took all of these primary endothelial cells in culture, treated them with this same drug, and asked, does their transcriptome shift in the same way that our Notch heterozygous patient endothelial cells shifted? And in fact, that was the case when you look broadly at RNA-seq. I'm just showing you here the changes in those 3 key transcription factors, SOX7, TCF4, and SMAD1, that we think are the main drivers of this switch. And I think in each case, which you can appreciate, is both in patients with tricuspid valve, 3-leaflet valves, as well as 2-leaflet valves. In all of these, most of these patients, each of these transcription factors was similarly upregulated, as you can see here in orange, in each case, and the levels were restored back to normal by this drug in every one of these cases. So that gives us a lot of hope that this drug will be useful in the broader population, regardless of the genetic etiology in this setting. And so I think we're ready now to take this drug and move it into clinical trials and see if this can be the first medical therapy available for patients with this disease that has no other treatment other than cutting the chest opener, in some cases a catheter-mediated delivery. So, Jeff, what is the time here? I don't have my watch with me.
You can go as long as you want. It's 12:32 right now. You've been going about an hour. About an hour.
Let me stop here. I had one other story that I thought I likely wouldn't get to, but that has more to do with oligogenic inheritance of disease. But I think for the sake of time, we're probably better off stopping at this point. And I think what I hope to share with you today is our approaches of, if I can get rid of this. How we, with our approach, we've been able to leverage these developmental networks both for regenerative medicine, but also in uncovering the mechanisms underlying human genetic disease, and then combining that with IPS technology to actually come to a new therapeutic. And so these are the folks in the lab, our current members of the lab. I try to mention some of the folks that were in the lab before that contributed some of this work, and our wonderful collaborators at Gladstone and beyond. With that, here's our current lab. I'll stop, and if we have time, I'll be happy to take some questions.
Yeah, we have plenty of time. Wow, thank you so much for that. I'm going to say that was an amazing talk. In the chat, I put my question. Early on in your talk, you suggested that Moderna-Pfizer approach. Are you suggesting that using open heart surgery, you could reprogram fibrotic tissue into cardiac muscle using the holy trinity you identified, or others?
So I think the modified RNA approach that Moderna and Pfizer have used may be most beneficial for the condition where we have the cell cycle regulators, where we only want those to be around for 3 or 4 days. And the challenge usually when using modified RNA is that if you want longer lasting expression, you don't get that. It's only a couple of days. And so it's limited in its utility. For the cell cycle regulators, it's perfect. That's exactly what we want. We want to be there and gone. So that's where I think it'd be most useful for the reprogramming of the fibrotic scar tissue. You know, we used to think that wouldn't be very useful because the cell biology actually takes several weeks to convert. But in fact, I didn't mention this from our mechanism studies using ATAC-seq and the part that I showed you how that epigenetic shift occurs. But I think I didn't mention is, much to our surprise, all of that epigenetic shift occurs within 2 or 3 days, which was a huge surprise to us. And then the cell biology follows for weeks, but in fact, all the action is really quick. So you know, it's worth trying now that if we can actually deliver these, it's once again worth trying if we can deliver it with that, because that, again, with modified RNA, would be cleaner and not involve gene therapy, which still has a number of hurdles to it.
So it requires open heart surgery, right?
Yeah.
The stenosis story resonates with me. My father just went through that surgery. He had it delivered, his new valve, his femoral artery, or through his leg. Amazing. He's 87. And when he had the surgery, and 1.5 years later, he's as right as rain.
That is great. It's been a real technological leap. It used to be that in people of that age, we actually wouldn't do open heart surgery because the risk was so high. And so this new catheter-mediated delivery was initially approved for people of that age group. And now it's such a good operation that it's going down in lower ages. And so that's, I think, a really good intervention as well. What we're hoping is that in the future, we'll be able to, cardiologists often detect it before people need surgery, and then we just watch it over years until it's bad enough to do surgery. And my hope is that with this, we'll just stave it off, even if we slow it down. You don't have to stop it. You slow it down, then that's probably enough.
Beautiful. Didn't Mick Jagger just
What's
have
that?
that?
Yeah, Mick Jagger has had it. Arnold Schwarzenegger had it when he was 50, even earlier. So yeah, it's very common.
Okay, more questions from the group?
I see one in the chat, but Doug, if you want to go ahead and ask that, would be fine too.
Oh, so my question is, it's fascinating that changing telomere length modified the way Notch interacts with downstream components to cause calcification. Is it, why? What do you know about that? Is it telomere length, or is it the RNA that was knocked out, or do you have any idea at all?
Yeah, it's a great question, and one I think we have a unique model system in which to answer a question that's still unanswered in the whole telomere field. So that, as you know, there's a connection between telomere length and aging. But in fact, when we came upon this, I went to Liz Blackburn, who's on our campus and won the Nobel Prize for telomeres, and I was a bit surprised since I didn't really know that field so well. She said, we still don't have any idea the mechanism by which shorter telomeres are connected to aging. And so she was actually really excited that maybe we could use this. And so there's one hypothesis that we're testing right now that I don't know if it's true or not, but telomeres have been thought to now loop back to contact promoters in the chromosome and affect their transcription. And so one possibility is that by shortening them, you affect that ability to loop back and affect transcription at a certain site. So we've looked at that and where telomeres actually contact by high C in the normal state in 3 dimensions. And there is an enrichment of the genes that are dysregulated that are normally contacted by telomeres, but that's just correlative. And we're trying to now do experiments that I'm hopeful with single cell high C approaches will, for the first time, be able to get at that. The technology hasn't really been there to answer it yet, but we are actually now able to do that experiment. We're trying to do that. So understand the mechanism, I think, would be fascinating, but we don't right now.
Student Dr. Clement, go ahead. You're muted.
Thank you for speaking with us. That was a great talk. I was just asking, I know that there are efforts to create sort of scaffolds to go over dead heart tissue that are implanted with the cells derived from the patient's actual heart cells as a way to replace dead heart tissue and perhaps, restore, the heart function. I'm just wondering if you see, as that progresses and as Gladstone's drug candidates and maybe even mRNA candidates, do you see sort of a synergy going together with those 2 modalities?
It could be, and I'm glad several groups are pursuing either direct transplant of stem cell derived cardiomyocytes or built on a scaffold and trying to encourage engraftment. So a number of investigators are trying those and are moving those forward with various other companies, and I think that's a really good thing because that may work. As I mentioned, some of the monkey studies that have been done now are encouraging, but they've had trouble connecting electrically, and at least early after transplantation of those cells, they've had a lot of rhythm problems that actually, if you get through that in monkeys, at least seems to resolve, maybe perhaps as the myocytes mature and engraft better. So I think it's promising, and I'm eager for those to proceed and multiple modalities, because each one actually is really difficult and has many reasons why they'll fail. And there's such a need. Jeff, you mentioned your father. My father suffered from heart failure for several decades and finally succumbed to that about 2 years ago. And it was so disappointing to, we'd never had any really disease-altering therapy for it. We just mitigated the damage that was there pretty ineffectively. So there's a great need. It's still the #1 killer around the world. And so I hope all those get move forward. And if more than one of them could work, I think that'd be fabulous.
Go ahead, Dr. Wolf.
I was going to joke that you're kind of making the silk purse out of the sow's ear. I mean, it used to always, you can't make a silk purse out of a sow's ear. And now it is changing. But my question was, after you were able to cause the transition, do the fibroblasts now come in? I mean, you did lose some cells there with the death. So are the cells numbers repopulating?
Yeah, the cardiomyocyte numbers repopulate, and fibroblasts, one may be concerned that fibroblasts are important also, and if we converted so many of them that we might deplete those, that could be negative. In fact, we're not good enough to do that. So only maybe in the best case scenario is 15 to 20% convert. But if, let's say we were good enough and the fibroblasts, unlike the myocytes, actually can re-enter the cell cycle and make more of themselves. So
yeah, that was my question, is whether or not, if you got conversion, did the fibroblasts repopulate so that you were actually regenerating your number of cells that you started off with to begin with?
Yeah, we haven't looked at that rigorously, whether they do or not, but we certainly have every reason to think they'd be capable of, because since they are not post-mitotic.
Yeah, I'll plug my father one more time. Former chair of biology at Nebraska Wesleyan. Now that he's survived his heart surgery, he just sits around and mutters about his telomere length. Are my telomere? I know they're short. So fun. So, are there any more questions? if not, let me say here, such a stunning and interesting talk. I want to thank you so much for joining us and bringing this to KCU. And again, it's okay that I recorded it and posted. You can go head to head with.
Yeah, yeah, that's fine. I shared some unpublished stuff, but I don't really care. I tell everybody everything.
Science in the public interest.
Exactly.
I love it. I love it. Thank you. Thank you. Everybody unmute and thank our speaker.
Thank you. Thank you. Thanks for inviting me, Jeff. It's great to see you again. And congratulations on all the stuff you guys are doing there.
Appreciate you, brother. I do.
Take care.
Have a great day.
Bye-bye.